Hello, 7th grade. It's Miss Odell here, and we're going to be reading through and working through Chapter 6A, which is titled Bacteria. You familiarized yourself with that by reading through and highlighting some bold words and some terms, so that will help you as we work through this, okay? Now, bacteria. Bacterial structure. Now, we're going to read through this, and then I put my own picture in because I want to maybe try to make it a little more clear. But bacteria are found in the air, in soil, in water. So I guess you could say, where is it not found, right? Anyway, bacteria. So the structures of bacteria. Well, it has a DNA. All right? So you'll see the DNA right in here. These little things there are considered the DNA, and I'm going to show you another picture here in a minute. I think it makes it easier for you to be able to identify. Many have one or more plasmids. Now, they are the small, often circular strands of DNA that are separate from the main chromosomes, but they're called plasmids. The next thing we'll talk about here is bacteria have ribosomes that produce proteins. Bacteria have a cell membrane, and many have a cell wall. Some bacteria has a capsule, and bacteria lacks nuclei, which is speaking of the nucleus, and a membrane-bound organelle. Okay? Now you can pause this at any time to take notes and so forth. But let's look at this specific diagram that I put on here, because I think it makes it easier for you to see the parts of the bacteria. First of all, I want you to take note on the cell membrane right here. That's like the inner part. And then the cell wall is even closer on the inside. This is referring to the capsule. Remember, the capsule is the slimy part. It's like a slimy coating that covers the cell walls. The capsule, remember, it prevents the bacterium from drying out. That's why it's wet. Endospores we don't really need to talk about right now, but the flagellum are these little strings that sort of hang out the bottom, I guess you would say. Plasmid, here's your plasmid. Here is the chromosomes. It's also talked about the ribosomes. And then, of course, we have our cytoplasm. All right? Guys, this is a short chapter, and it's got a lot of complicating stuff in it. So if we can just try to grasp as much as we can, in the next few chapters, it will become more clear to you. All right? Moving forward here, there are various bacteria types. In the past, last year in science, we talked about archaebacteria, if you remember. But archaea are quite different from bacteria. There are only a few hundred recognized species. Okay, so let me read this to you here that's in your textbook on page 122. It talks about the hot rainbow-colored waters in the Grand Prismatic Spring of Yellowstone Park. Shown on this page, spread would be deadly for human. But they are teeming with microscope life. Many bacteria lives there. The water becomes cooler as it moves away from the center, and the bacteria living in the different temperature areas of the stream produce the different colors. I think that's fascinating. And they share the water with other prokaryotes classified in the kingdom of archaebacteria. Although like bacteria in many ways, archaea are quite different. Their cell walls lack a molecule that, let me try to pronounce this for you, peptidoglycan. That is found in bacteria, and although their genes are more similar to eukaryotes, than the bacteria, although Archaebacteria are most famous for living extreme places, such as the hot springs, like in the swamps, and even in the human body. It shows you some different shapes of the bacteria on that page if you get a chance to take a look at that on page 122. Archaebacteria live in small groups and only a few hundred recognized species. All right. We talk about the shape here, the spherical, rod shape, and spiral shape. That's the three basic shapes of the archa. So, before we get to the reproduction, let me just ask you a few questions here. How many cells do you think your body is made of? Well, our textbook tells me that it's typically made of about 1 trillion or so cells. How many microbiota cells do you think live in your body? Now, these are extremely micro, would let you know that it's extremely small. And the cells are about the same as the number of our human cells. You don't have to write this down. This is just some extra information for you, okay? Now, bacterial reproduction. Of course, you know it has to reproduce. And this is on page 123. Now, like all living organisms, bacteria has to reproduce. Normally, bacteria reproduce by binary fission. You see that right here. Which the DNA in the chromosomes and any other plasmids is replicated. They become duplicated and replicated there. The cell membrane that pinches together to form two bacterial cells. The two bacterial cells are identical, as you see here. They, you know how you see here, they can be pinched together here, and then they produce two identical bacteria cells. And our textbook tells us that this can happen about every 20 minutes. So let's think about this. In 20 minutes, one bacterium can produce two bacterium. In another 20 minutes, two bacterium divide to produce a total of 4. So in an hour, one bacterium can become 8 bacteria. If this is continued, at the end of a day, one bacterium will become nearly 5 sextillion. That's 5 followed by 21 zeros. So bacteria, a lump of bacteria that large enough that would weigh 4.5 metric tons. So the bacteria formed are identical, and some can reproduce in as little as 20 minutes under ideal conditions. Okay? So an endospore. What is an endospore? Well, I'm going to write some of this information in here for you, okay? All right, so an endospore. Now, sometimes the conditions are so poor that bacteria struggles to survive. We've read about that in the past, but some bacteria escape these conditions by forming endospores. So the bacterium's DNA and ribosome are enclosed in a tough case that can survive heat, cold, and a host of other different conditions. Okay, so endospores is a bacterium's DNA and ribosomes. They become enclosed in a case, and that process happens in order for it to survive any unfavorable conditions or any harsh conditions that it may encounter. Okay? In some cases, actually, endospores can survive thousands of years. Anyway, we'll talk about later on when we get back into the classroom. We'll talk about actually growing some bacteria in a petri dish. Probably, well, we'll get into that later, but we'll probably do that when we get back into the classroom, which will help you to understand that as well. All right, moving on. So, bacteria in the environment. Some roles of bacteria include many of our bacteria in your body keep other less friendly bacteria from starting to grow, so they're not all bad. Others contribute to your health. Bacteria fix nitrogen in enabling plants to use it. If you will open your textbook with me on page 124, you will see bacteria in their environment. Bacteria are so common that it's hard to find a process that they are not involved in. So if you look at this with me, you will see that as you read through the chapter, you have as many non-human cells as human cells. Okay, we'll move on here. So plants need nitrogen to grow, but they can't use nitrogen gas in the atmosphere. So instead, the atmospheric nitrogen must be transmitted into forms that plants can use. Now, bacteria can live in the soil and are very important in fixing that nitrogen. So some of the bacteria live in the roots of plants such as beans and clover. That's why they call it bacteria fix nitrogen, because the plants use it. So bacteria in ruminants help those animals digest cellulose. If you look at number 3 on page 124, it's in the earth cellulose. Certain mammals called ruminants have a special chamber in their stomach, and that content contains the bacteria, and that bacteria actually breaks down the cellulose into other nutrients that they can digest. Sounds a little gross, but it's just part of life, okay? Bacteria are essential for making many foods. If you flip over to the next page, plants and animals use material from their environments to build their body systems. If these nutrients remain trapped in their bodies after they died, the world would run out of nutrients. So we need lots of that nutrient to survive. Not all bacteria are beneficial today. Some is not. Bacteria were created very good, but the curse included bacteria becoming the cause of certain diseases. Bacteria bacteria is important for making cheese, green olives, yogurt, pickles, salami, and things like that. So, let me answer a few questions here, ask you a few questions. So the soil, let me just tell you this. There are some very important ways that the world would be different if bacteria did not exist. Some of those ways is that we would not be able to digest food as well. The soil would not be as rich for growing plants. Many animals would not be able to get nutrients from the cellulose. We wouldn't have certain foods such as cheese, green olives, yogurt, pickles, and salamis. And I don't know about you guys, but I sure love my cheese and pickles. And our risk of sickness or infection from bacteria would be absent. So there we have it. Now, moving on. Let's talk about some questions here that you may have. So, place the steps of binary fission in order. Let's move on, skip that. Okay, here's the answer to it. Well, one bacterium, then DNA duplicates, cell membrane pinches in two, and then you have two identical. That's talking about the reproduction, okay? All right, so what, name one of the differences between prokaryotes in kingdom Archaea and those in kingdom Bacteria. Well, remember, Archaea lacks a certain molecule in their cell walls, and they also have genes that are more similar in those in the eukaryotes, okay? You don't have to write that down. That's just information. All right, so that ends our lesson for today. Let me know if you have any questions. I know this is a very complex lesson, but I promise you it will become clearer as we continue to work through it, so don't panic, okay? Have a great Tuesday, guys.